Hi all, welcome to CSEC Biology with Mr. Charles. Under the topic of transport and regulation in humans, today we will be looking at the immune system and human immune response. By the end of this lesson, you should be able to answer the following objectives. 1. State how the human body defends itself against pathogens. 2. Describe the role of blood in defending the body and in developing natural immunity. 3. Explain how the principles of immunization are used in disease control. And 4. Discuss how vaccines are used to provide artificial immunity. The body's natural defense mechanisms include both external and internal aspects. The external defenses prevent pathogens from entering the body. Pathogens are disease-causing elements. On the other hand, internal defenses operate within the organism to kill pathogens that have somehow managed to enter. The immune system consists of the skin, parts of the respiratory system, and parts of the digestive system, as well as the blood. Let us look at the roles of the main components of the immune system. First of all, along the respiratory tract, there are stratified columnar ciliated epithelial cells. These membrane cells, they have goblet cells within them that produce mucus, and the mucus helps trap particles and bacteria. The cilia creates a wave motion that sweeps mucus along. In the stomach, glands in the stomach produce hydrochloric acid that kills bacteria that may be ingested together with food. In the cervix, the female cervix, a plug of mucus acts as a barrier to microorganisms so that this area is kept sterile. Okay, the uterus is kept sterile. We also have the skin, which forms a physical barrier. When the skin is broken, blood clots at the opening and a scab forms to stop pathogens entering the body. The sebaceous glands produce sebum, okay, which is an oily substance which kills bacteria and fungi. We also have tears from the tear gland that contain lysozyme. Lysozyme is an enzyme that destroys bacteria. Then we have the white blood cells of the blood, which are produced in the bone marrow of long bones and lymph glands. These white blood cells destroy bacteria and viruses. Okay, these are the roles of the main components of the immune system. This slide looks at immunity and immune response. Immunity is an individual's resistance to a specific condition or disease, or an individual body's response or resistance rather to a specific condition or disease. Now, when a person's internal defenses destroy a specific pathogen, that person is described as having an immune system that's immune, or that person is described as being immune to the pathogen that caused the disease, or immune to the disease. Such resistance may be short-lived or long-lived. Now, the immune response is the body's or the organism's reaction to being invaded by foreign material. Okay, the foreign materials could be viruses, bacteria, toxins, or even other unrecognized proteins. Now, all of those substances are identified as foreign by the human body. Okay? Now, let's continue to look at immunity and the immune response. First of all, we have antigens. And antigens are cell surface markers. Okay, they are cell surface markers because they are found on the surface, on the surfaces of cells. And they are able to provoke an immune response. Now, once the antigens are identified, okay, as part of the immune response, the lymphocytes would now come in and produce antibodies that would bind to the antigens and destroy them. Now, it is important to note that each antibody is specific to its antigen, which means that it can only bind to that particular antigen. For example, the antibody that acts against typhoid or the typhoid bacteria will not attack the pneumonia bacteria, okay, because the antigen is of a particular shape that allows the antibody with a particular shape to bind with it. Okay, so the antibody is specific to one antigen. Okay, also important to notice that the body is able to produce tens of millions of antibodies during a lifetime. Let's look at natural immunity. Now, antibodies stay in the body for some time after they are produced, okay? And that's important in case there is reinfection, okay? So in case there is a reinfection, the antibodies will look around to attack the same microorganism should they show up again. Now, if the antibodies do not remain, the antigens on the surface of the microorganism or microbe is another word for microorganism. These antigens are identified by the lymphocytes, okay, on further attacks. Now, because of that quick identification, the lymphocyte produces larger quantities of antibodies than the first time around, making the immune response even faster than the initial response. Okay, and in that case, we say the body becomes resistant to the disease. That is why persons rarely contract or get infected with diseases like chickenpox and measles more than once, because once there is a first infection, the body memorizes, or the body's immune system memorizes the antigens that were presented on the bacteria that caused such infection. And in case there is a second infection, okay, even though the bacteria shows up itself a second time, the response is faster and more effective. Okay, so there is hardly a second contraction of the disease. Okay, so in the previous slide we started explaining natural immunity, and in this current slide we will continue to explain natural immunity. However, we will be doing that through diagrammatic illustrations or analysis of diagrammatic illustration. Now, there are two aspects to natural immunity. There is what you call the primary response. Okay, primary response, and I need my pen here because I need to ensure that you understand this very well. There is a primary response, and then we have the secondary response. If you look to the left of the screen, you would see we have a red graph and a blue graph. Okay, this one, the red graph represents the primary response. Let me write, well, I wouldn't write it, but this is a primary response. Okay, and this is the secondary response here. Okay, even to the right of the screen, we have primary response, secondary response. So we must recognize that natural immunity operates in two phases. Okay, now when an organism becomes initially infected with a pathogen, it has its first exposure. Okay, so I'm not sure you're able to see this here, but in this red box we have antigen first exposure. Okay, antigen first exposure. So this is the organism's first exposure to the antigen. Okay, this arrow here tells us that because this is the first exposure of the organism to the antigen, okay, and because the immune response would be very slow, it is inevitable that the individual would get the disease. Okay, that would be the outcome of the exposure. Now this arrow here tells us that even though there is disease, okay, resulting from the first exposure, there is a specific immune response. And please pay attention to the word specific, because remember in the previous slide I stated that each antibody is specific to a particular antigen. So you have a specific immune response. Okay, and even to go further in that explanation, that's why it is important when you're taking antibiotics that the antibiotics you are taking are specifically created for the bacterial infection you are trying to eliminate, because the antibiotic for one bacteria would not work for another bacteria. That's why it is strictly advised that before you take any antibiotic, you should have been prescribed to do so by a medical doctor. So you have your initial exposure to the antigen, okay, and although you get the disease, there is a specific immune response, okay, resulting in an immune reaction, and that the main or the ultimate goal of that immune reaction or response is to destroy the antigen. Okay, and that would involve lymphocytes that would produce antibodies that would bind to the antigens. Okay, pay close attention. The lymphocytes would produce antibodies to bind to the antigens. The antigen antibody complex would then clump with other antibody antigen complexes, okay, in a process called agglutination. Let me write it here, AGG. Okay, all the antibody antigen complexes would clump together in a process called agglutination. And in that process of the main purpose of that process of agglutination is to bring all the foreign elements in one place, so that the phagocytes, remember you have lymphocytes and phagocytes in blood. Okay, remember one of our objectives was to explain how white blood cells are, how blood is important in the fight against infections. Well, the lymphocytes and the phagocytes are present in blood. So when the lymphocytes produce the antibodies bind to the antigen. The antibody-antigen complex then clump with other antibody-antigen complexes in a process called agglutination. Okay, this long word I've just written here. And agglutination helps bring all the antigens, okay, all the foreign particles in one area so that the lymphocytes, which is the next part of the immune system or the white blood cells, to extend pseudopodia around the clump, right, resulting in the lymphocyte eating that clump. Okay, so pseudopodia, PS, and pseudopodia basically means fake feet. Okay, oops. Okay, pseudopodia. Pseudo means fake, podia means feet. So they feed by extending pseudopodia around the clump, and then after the clump is completely surrounded, these macrophages or lymphocytes would secrete enzymes from vacuoles on the inside to destroy the pathogen, okay, within its membrane. Okay, good. So this is the immune reaction. So when we talk about immune reaction, we are talking about the involvement of the lymphocyte and the involvement of the phagocyte. Okay, and both the lymphocyte and the phagocyte works together in the immune reaction to destroy the antigen. When all the antigens are destroyed with the pathogens that carry them, then you have recovery. Now, in this primary response, this immune reaction occurs very slowly because it takes a lot of time, okay, to produce the antibodies after recognition of the antigen. Because remember, this is the first exposure. There is absolutely no antigen in the blood to fight against that particular, there is no antibody rather in the blood to fight against that particular antigen. So the process of creating the antibody has to start, okay, from scratch, and it has to take a long process to occur. So the immune reaction is very slow. Okay, so it will take a longer time to recuperate or recover. Now, if you look at this arrow here, you would realize that there is a specific immune state. Okay, so because of that immune response, there is a temporary immunity that results. Now, this temporary immunity may last for many years, and it involves memory cells that keeps record of past infection. So when you are infected with the antigen, the body's immune system memorizes that antigen. So the next time there is an infection, or if there is a reinfection or a resurfacing of the antigen, then the response would be faster. The recognition of the antigen would be faster, okay? And recovery would be faster, even to the extent that you may not even get the disease, okay? You may not even get the disease. So how this works is that when you have a second exposure to the antigen, when there is a second exposure to the antigen, okay, almost immediately there is an immune reaction. There is an immune reaction, and the memory, okay, the specific memory and recognition of the antigen, okay, because of the memory cells from the primary response, okay, it provokes rapid immune response. Okay, so almost immediately you get an immune reaction that is very rapid, and the destruction of the antigen occurs very quickly. Okay, so you have what you call a specific immune state, and then there is no disease. Okay, and as I stated in the previous slide, that is why very rarely you have persons being infected with things like chickenpox and measles, okay, more than once, okay, because of this natural immunity mechanism. Now if you look to the left, okay, you would notice that here the primary response is slow. Okay, it's slow. Look at how long it takes to start. Okay, how long it takes for the level of antibodies in the blood to rise. Okay, time, this is your x-axis here. Okay, this is your x-axis, this is your y-axis. Look at the time it takes before you start having the level of antibodies in the blood increasing rapidly. Okay, in this region here, the level of antibodies in the blood is increasing rapidly. Look how long it took for the level of antibody in the blood to increase rapidly. Okay, compared to the blue graph on the left. Okay, you have almost no time at all. Okay, so when during the second infection, you have a quicker response because you take less time to start producing antibodies, okay, rapidly. Okay, and the secondary response results in the production or the generation of much more antibodies than the primary response. Look at the y-axis. Look at the level of antibodies for the secondary response. Look how high it is. Compare that to the highest level of antibodies in the primary response. Look how low it is. Okay, so you have this huge difference. You have this huge difference in the level of antibody in the blood. Okay, so the secondary response is much more effective than the primary response. Okay, both in the level of antibodies present in the blood during the secondary response compared to the primary response, and in the time taken to generate a response. Okay, this here represents the time taken to generate a response. Okay, it's called the lag phase. Let me write it here. Time taken to generate a response. Okay, and this time taken to generate a response is called the lag phase, LAG, and that is what I am circling here. Okay, that's what I'm circling here. Okay, so make sure you study this diagram carefully and look at the explanation on the previous slide. Okay, the explanation on the left here and the explanation on this slide. Okay, so be mindful of that, because this mechanism of natural immunity is very important. Okay, and the basis of quicker natural immune response is the specific immune state, or the memory cells from the primary response that make the secondary response more rapid and almost immediate, okay, resulting in no disease. Great, very good. Okay, so what we've just explained, as I stated, is natural immunity. Now we're going to look at artificial immunity. So this slide is designated to the explanation of immunization and control of disease. Okay, so how is immunization used to control disease. So what is immunization? Immunization is a process by which a person becomes protected from a disease or a condition through vaccination. Okay, so vaccination leads to immunization. Okay, vaccination leads to immunization. Immunization is a process of becoming protected. Immunization is as is rightly stated down here, vaccination is so immunization is a process by which a person becomes protected through vaccination, and vaccination is the act of introducing a vaccine into the body, okay, to produce immunity to a specific disease. So vaccination is a process by which the vaccine is introduced into the body. Immunization is a process of becoming protected. Okay, now the injected substance is called the vaccine, right? Okay, so what is the vaccine that is introduced into the body, okay, is the injected substance. Now that it may not always be injected, because some vaccines are introduced into the body orally. Okay, so vaccines may be given orally or through the mouth or intravenously through an injection, okay, into your blood vessel. So it's not always intravenous, it can be through your mouth. Okay, now if it's an antibody, then taking it orally would not make sense, because there is pepsin in the stomach that digests proteins. So if you take a vaccine made from antibodies, then it would be digested in your stomach to amino acids and so forth, and that would make it that would destroy the vaccine and not make it effective anymore. So antibodies are injected. Okay, so if it's another type of vaccine, then it would be given orally. So some vaccines are given orally, some are given intravenously. Now, immunization may promote active immunity or passive immunity to a disease-carrying organism. Okay, so keep that in mind. Good. Now, let us look at what vaccines are made of. Okay, vaccines are made from one of three things. Okay, so vaccines are either made from dead microbes. Okay, dead microbes. Example, whooping cough vaccines is made from dead microorganisms, dead bacteria. You could also use a weakened form of the microbe, which is harmless, and those vaccines that are made like these are called attenuated vaccines. Okay, attenuated vaccines, okay, containing a weaker form or a mild form. Okay, weakened also means mild. So a weakened or mild form of the microbe, okay, can be used to make the vaccine, and in that case it is called an attenuated vaccine. Okay, the tuberculosis vaccine is an attenuated one. You could also use a substance from the disease-causing microorganism. Okay, a substance from the disease-causing microorganism which does not itself cause the disease, okay, but it contains antigen that can be used to fight against the disease. Okay, and for example, the diphtheria vaccine is made from a substance from the disease causing bacteria that causes this diphtheria. Okay, genetic engineering, okay, is also used to produce new vaccines. Okay, So for instance, a gene from one organism can be incorporated into the DNA of another. Okay, and that can be used to produce new vaccines that can be incorporated into humans to provide artificial immunity. Okay, great. Now, there are two types of vaccines, and we're going to look at how vaccines work. So there are two types of vaccines. One type of vaccine provides active immunity, while the other one provides passive immunity. And I stated that in the previous slide. Okay, in this first slide on immunization and disease control, where I stated that immunization can promote both active or passive immunity to the disease-carrying microorganism. So, the two types of vaccines, one type provides active immunity, whereas the other type provides passive immunity. Great? So keep that in mind. Okay? Okay, so we have already established that immunization works or vaccines work by providing active or passive immunity, and this active or passive immunity are two types of artificial immunity. Let me get a different color ink. Use black. Good. Okay, so to the right of the screen we have a diagram that summarizes active and passive immunity. Now, active immunity is acquired when antigens, okay, when antigens are given as vaccines, okay, and this stimulates the production of antibodies which remain for a longer time in the blood. Now, those antibodies that remain would eliminate microorganisms in case of a future infection. Okay, so in other words, in active immunity, the person is vaccinated with antigens. The antigen stimulates the production of antibodies. Okay, the antibodies remain for a longer time in the blood, and when they remain, they are able to eliminate microorganisms or microbes in the event that there is a future infection with the same pathogen. Okay, so the vaccine for measles, okay, tuberculosis, polio, diphtheria, and whooping cough are all based on active immunity. Okay, all of them are based on the principle of active immunity. So keep that in mind. Passive immunity, on the other hand, is acquired when antibodies. Okay, let me get a different color ink. Passive immunity is acquired when antibodies are given as vaccine. Okay, and these antibodies are produced by other organisms. Okay, so they are produced by other organisms in response to a specific antigen. They are extracted from those organisms and given as vaccines. In a previous slide, I spoke of genetic engineering, where I said that genetic engineering may be used to produce new vaccines. Okay, those vaccines produced by genetic engineering are primarily based on antibodies. Okay, let me just write that here. Okay, so genetic engineering is used for the production of antibodies on a large scale, okay, in smaller organisms like bacteria. Okay, and when they are produced on a large scale, then they are extracted from the bacteria, purified, and then introduced into humans as vaccine, okay, because they have proven to work efficiently against that specific type of pathogen in the organism from which they were taken. Okay, so they're taken from the organism that's immune to it. They are mass produced in bacteria, okay, and after mass production they are extracted from the bacteria and purified, and after purification they are used as vaccines for humans, okay, antibody vaccines, okay, and these remember antibodies are proteins, so these vaccines cannot be taken through the mouth, they must be injected intravenously, okay. So these type of vaccines provide what we call passive immunity, okay, passive immunity, okay. So because you're introducing vaccines with antibodies that are already made, okay, ready-made antibodies, the body of the host is not making the antibodies, as opposed to active immunity where the patient is receiving the antigen, so that person's lymphocytes have to make its own antibodies with given the antigens it has been given, okay, with the antigens based on the antigen that it has been given. However, with passive immunity, the antibodies are already made, okay. So this is a key difference between active immunity and passive immunity. Now, passive immunity is also used when there is exposure to antigen but no immunity. So in cases like that, although the antigen is in the bloodstream, there is no immunity, which means that the antibody or the body cannot produce antibody against the antigen, okay, even though there is exposure, internal exposure to the antigen. So in that case, you would have to inject the person with antibody because it's not capable of making its own antibody even when the antigen is introduced into the patient. For example, pregnant women who are exposed to German measles, okay, German measles, may be given a vaccine of human serum, okay, or with antibodies from human serum, okay. So pay close attention to that example, okay. German measles have no effect on the pregnant woman, however it can terminate the developing fetus. Tetanus injections are also made from antibodies, okay. Tetanus injection are also made from antibodies, okay. Passive immunity is important for babies mostly, okay, since they are unable of making their own antibodies, okay. Remember we established that up here, exposure to the antigen, however the individual is not capable of making its own antibodies, so mainly babies, okay, because when babies are born they are not capable of making their own antibodies, they rely on the antibody from the milk produced in the mother's breast, okay, the mother's milk. So although those antibodies are short-lived, the length of time they are available to the infant for is sufficient to allow for that infant to be able to make its own antibody. So by the time that antibody wears off, then the baby would be able to make his or her own antibodies. Okay, so passive immunity is most important for babies, okay, since they are unable of making their own antibodies. Okay, so know the difference between active immunity and passive immunity. Great. So that was the last slide. Thank you very much for paying attention to my presentation. Please listen the video or look at the video more than once to ensure that you understand the content that is presented. Key slides are slide number 5. Okay, this slide that speaks of the roles of the main components of the immune system. We also have this slide here, slide number 3, which talks about external defenses and internal defenses. Then we have slide number 9 with the diagrams on the body's immune response, okay, which elaborates on natural immunity, the primary response and the secondary response, know the difference, and also be able to explain how the primary response influences the secondary response. Okay, and also last but not least, the final slide is very important. You must know the difference, be able to explain the difference between active immunity and passive immunity. And before I end this video, I will make mention of the objectives. Remember the objectives. Okay, it will do you very well if you are able to use objectives as questions and answer the objectives. Okay, write answers to each objectives so that you would be able to better understand the content presented during this video lesson. Okay, so that's it for me. Thank you very much for paying attention and listening to this video this far. It has been a pleasure. Okay, do enjoy the rest of your day.